So, hello everyone. First of all, thank you to the organizers for this wonderful opportunity to present here tonight. And today, my name is Ihor. I'm a master's student at the University of Sherbrooke, and today I'm going to present my project, which is called Multi-omic Analysis of Testes Reveals Pseudogenes as Overlooked Biological Actors. But first of all, let's talk about pseudogenes. So, what are they exactly? In fact, they're just copies of protein coding genes, which may arise in different ways in our genomes, but their common characteristic is their loss of ability to produce a functional protein. Thus, in the literature, they sometimes are referred to as junk DNA. However, a growing body of evidence suggests that pseudogenes can actually be transcribed. Moreover, pseudogene expression can be specific to tissue subtypes. Like, for example, in this 2014 paper, the authors showed that pseudogene expression was specific to tumor subtypes in 7 different human cancers. So in our project, we hypothesized that pseudogene expression can be specific to cell types and or cell states as well. And to test this hypothesis, we performed the single-cell RNA sequencing experiment on 4 testes of adult mice. And after bioinformatics analysis of this data, we expected to observe cell types which would be defined by pseudogene expression. And before you ask, I'll answer right away. We decided to choose testis tissue because we know, as a matter of fact, that testes express a lot of pseudogenes. However, analyzing the sequencing data for pseudogene expression right away would be rather complicated. In fact, most pseudogenes have on average 85.5% of sequence identity with their parental genes. This gives rise to so-called multi-mapping reads, or MMRs in short, during sequencing data alignment to the reference genome. These are reads that align equally well to multiple genomic regions. Thus, to observe pseudogene expression in single-cell RNA sequencing data, some method for handling multi-mapped reads should be applied. So for aligning our data to the reference genome, we used StarSolo tool, which has 4 different algorithms for MMR handling implemented in it. And based on the sequence coverage uniformity, we decided to choose expectation maximization, or EM, algorithm for handling multi-mapped reads. To illustrate what I mean exactly, I will show you an example of a pseudogene which was rescued by EM algorithm. So here you can see the sequence coverage plot for this particular pseudogene before any MMR handling and filtering. This means that all uniquely mapped reads and all multi-mapped reads are still present on this graph. But for this particular pseudogene, the situation is even more interesting. In fact, it is fully contained in a long non-coding RNA gene. Thus, there is no theoretical possibility for this pseudogene to have any uniquely mapped reads. However, it's worth mentioning that when we look at this graph, all the uniquely mapped reads to the long non-coding RNA gene, they are located somewhere around here. However, based on the sequence coverage, even though all of those are multi-mapped reads, it's reasonable to assume that pseudogene is also expressed in our data. And indeed, EM algorithm was able to rescue some reads to this particular pseudogene. So before analyzing our data in more detail, we decided to look at how many pseudogenes we actually detected. After performing quality control and normalizing our expression data, for any gene or pseudogene to be confidently detected, it needed to have at least 10 normalized counts in at least 2 samples. And as you can see on this stacked bar chart, where every stack represents a particular biotype, pseudogenes comprised approximately 7% of all detected biotypes in the data. And as you can see on these violin plots of the average normalized read counts in a logarithmic scale for each biotype, pseudogene expression levels were significant and comparable to the other biotypes in the dataset. Next, we performed downstream bioinformatics analysis of our data, including dimensionality reduction, clustering, and cell type annotation with RescueSC package developed earlier in our laboratory. And we were able to identify clusters with different cell types ranging from spermatogonia and different developmental stages of spermatocytes up to Leydig cells and Sertoli cells. Moreover, our data had on average 3,700 genes identified per cell, meaning that our sequencing data had quite a good quality. Finally, to observe cell type specificity of pseudogene expression in our data, we performed differential gene expression analysis of every cluster against all the other cells in the dataset. We identified 27 differentially expressed pseudogenes in particular cell types. However, what's also interesting here is that every cluster had between 4 and 7% of differentially expressed pseudogenes inside it. This shows basically the potential of pseudogene expression to influence cell type specificity in mouse testes. Now, to assess whether pseudogene expression can be specific not only to cell types but also to cell states, we performed the trajectory analysis of our data, and we identified 1,053 pseudogenes which changed their expression patterns through cell states. And among them, 21 were differentially expressed in specific cell types. There is some evidence in the literature that pseudogenes can not only be transcribed, but actually also translated. We wondered whether we could find evidence of translation of pseudogenes in mouse testes. And to answer this question, we performed the tandem mass spectrometry experiment on the same 4 mouse testes. And after analyzing our data with transproteomic pipeline using a custom database consisting of canonical proteins from Tremble and predicted pseudogenic proteins from OpenProt, we expected to see evidence for translation of some pseudogenes in mouse testes. And after analyzing our proteomics data, we were able to confidently identify 103 pseudogenic proteins detected with at least one unique peptide. 13 of them were also detected according to HPP guidelines, which means that they were detected with at least 2 unique non-nested peptides, being more or equal to 9 amino acids long. Also, they were not a result of a single amino acid variant of a canonical annotated protein. Moreover, 40 of those 103 pseudogenic proteins were as well confidently detected in our single-cell RNA sequencing data. There were 63 pseudogenic proteins which were detected in proteomics, but these pseudogenes were not detected in single-cell RNA-seq data. But those, in fact, were technically present in the RNA sequencing data, but their expression levels were below the thresholds for confident detection, which we discussed earlier. So to summarize all the major findings, I want to draw your attention to these Venn diagrams. So among the 21 pseudogenes which were differentially expressed in particular cell types and which were changing as a function of pseudotime, meaning that they were specific to cell states, 18 were also confidently detected in proteomics data, meaning that they had potential to be also translated and not only transcribed. It's worth to note that of those 18 pseudogenic proteins, 1 was detected according to HPP guidelines, and it was a protein translated from a pseudogene of tubulin beta 4b. It has 98.2% of sequence identity with its parental protein, and that's why interpros can predict the same functional domains both for original tubulin beta 4b and for its pseudogenic protein. This signifies the potential functional role of this pseudogenic protein in flagellar motility and in spermatogenesis. So in conclusion, I would like to say that single-cell RNA sequencing with handling multi-map reads helps to detect cell-specific transcription of pseudogenes in mouse testes. Also, data-dependent acquisition proteomics detects confident translation of pseudogenic proteins even without any enrichment. And finally, we found evidence for the potential functional role of pseudogenes in flagellar motility and in spermatogenesis. I would like to acknowledge the members of our laboratory and our collaborators for helping pursuing and generating ideas for the project. And one more time, thank you for the organizers of Great Lake Bioinformatics Conference, and thank you for having me here. And finally, thank you, dear audience, for your attention.
All right, we can take any questions.
Thank you for the interesting talk. In case you don't see us, I'm Pinyan from Brock University, also from Canada.
Hello.
I wonder if you try to, I guess the example you have in the end is a process pseudogene. Do you also have any example of non-process pseudogene?
Yes, thank you. That's a good question. So, well, just for a nice picture, I'll come back here. Well, to be honest, yes, most of the found pseudogenes were processed pseudogenes, but there were a couple of cases, I think just one or two cases of transcribed unitary pseudogenes and some unprocessed pseudogenes as well.
All right. So do you try to distinguish between active transcription versus passive transcription in those cases?
No, I don't think that we do it in this project.
I guess one of the example you showed seems to be because it's a part of a long coding RNA. So
Right.
I think that's a passive transcription as part of the host gene, right?
Maybe, but I'm not sure. I have to double check though. But it might be that the pseudogene of tubulin beta 4b is in the different chromosome. So it may be as well passive, but it could be also like a part of regulatory function. So obviously our project does not have enough information to answer this question, but further research may show it as well.
All right, thank you.
Thank you.
Hi, Ihor. So I want to follow up on those questions. How many of the genes, the pseudogenes you looked at, were randomly duplicated with their parental gene? Do you know that?
Thank you for your question. No, I'm not sure how many of those are unprocessed, which are results of segmental and tandem duplication, unfortunately.
Yeah, well, something you might look at, because if they
Sure.
are tandemly duplicated, then you can imagine they might be piggybacking on the same promoter.
Yeah, exactly. That's a very good point. Thank you.
I look forward to hearing the next version of your talk with that information.
Thanks. Sure.
Thank you for your talk.